and thanks for coming out to hear from us. I'm just going to tell you for a few minutes the kind of work I do to study how the brain works or doesn't work. And the key to my research is I study eye movements. And it's not that everybody makes crazy eye movements. We just use the measurement of the eye movements as a way to measure the behavior of an individual to see if there's a problem in the brain or not. And I hope you'll be able to appreciate where this is going to go as I talk. So as I walk around here, and I've got some slides up here, all of you are making eye movements right now. You suddenly moved your eyes over here, because I was over here. And if I walk back over here and wave my arms and talk, all of you moved your eyes to look over here. And the reason you're moving your eyes is because vision is so important to all of us as humans as a way to get information from the environment. And in the back of our eye is a small specialized part, right in the center of the eye, where you have the highest visual acuity. So all of you are looking at me right here. So if you all keep looking here, don't look away from this point, but I'm going to leave here. Keep looking over there. And now if I stand over here and start waving fingers and that, how many can see how many fingers I'm holding up? Probably none of you. Because you've got to turn and now look over here. How many fingers am I holding up? Now you can see right away. So what you had to do is move this small part of your eye. You're moving it around all the time, trying to follow me where I'm going. And I think I have a couple slides that are just going to demonstrate this behavior, what we're looking at. Imagine if on this whole screen there wasn't these words about my research, but just this picture was here, and you were looking at the picture. And if I was measuring your eye movements, this might be the pattern of eye movements we would measure from each of you when you were looking at this picture. And effectively, this is a dot-to-dot diagram, which is signifying where that small center part in the back of your eye was. And so what you can see here right away is the individual who made this pattern of eye movements was clearly looking at the two eyes, the nose, and the mouth of this pretty girl for most of the time. And sometimes they're looking around the outside of the face. And that's what you're doing every day, all day long. All of you do this about 200,000 times a day. It's probably the single most important, most numerous voluntary movement you make every day, all day long. And probably none of you think about what you're doing. You just take it for granted as your eyes dart around. And so what I'm just going to spend a couple minutes this evening is showing you how we can take this behavior and use it to investigate how the brain works. So I'm going to do it in a couple ways. I'm going to show you how we do it in the lab, and I'm going to show you some examples of the kind of data we collect from different patients. But I'm going to show you a little bit now the science of how this works and why we study eye movements. So this is just a view of the human brain, and there are just a number of small areas located all around in the brain, up in the cerebral cortex, in a place called the cerebellum, a couple areas down here in the brainstem. And there are small regions in all of these places that are playing an absolute critical role in that behavior of all of you moving your eyes around in this room right now as I speak. And the beauty of this slide just shows these areas are small, but they're scattered almost all over the brain. And what this means is, if we think about this from a patient, if there's almost anything wrong in the brain except the spinal cord. So eye movements are no good to study the spinal cord. But eye movements are very good to probe if there's anything wrong up here in the brain. So this becomes a very good tool to use to study problems in the brain, be they in children during normal development or abnormal development. To also use this approach in aging. Effectively, what's happening in aging, as we slow down and our brains are starting to fall apart, it's going to affect this behavior, and we can exploit that and use that and measure it to see if we can find biomarkers for disease. So that's basically what we're trying to do in my lab. And the next slide's going to overwhelm you. All of these little areas, I'm going to put them into a circuit diagram, and all of you are going to go, oh my God, he's going nuts here. Where's he going? Just pay attention to these colors. That the red colors here are kind of the parts down in the brainstem that are actually going to make the eyes move. If you want to look to the right versus the left, different things going down here. In green are the parts of the brain that are really doing the hard work for the visual system. So these are the visual areas. Up here in blue are the parts way up in the frontal cortex that allow us complex thought and make decisions about, you know, do I want to cross the street? Do I want to stay on the same side of the street? Do I want to go to Tim Hortons coffee? Would I rather get a Coke? These complex decisions involve these areas, including picking where you're going to move your eyes next. And other parts, and they're all like in this circuit. And so I'm afraid to put this next slide up, but I just will. For those of you that aren't brain aficionados, just pay attention to the colors, that's what matters. And there's only 4 colors here. There's blue, green, red, and orange. And basically, all I want to impress you with this slide is I just showed you that sweep of the brain. We actually know a lot about what's going on in all those little boxes that I showed you on that picture of the brain. And I won't go into that kind of detail tonight, but we know a lot about this circuit, and that's what we're going to use in my lab to study how the brain controls the behavior. Okay, so imagine we brought you to the lab, and maybe some of you will sit down at the eye tracker thereafter and just do a fake experiment. This might be what happened. Imagine you're looking at a big screen, so this rectangle's representing a big visual screen, everything else is in darkness, and you come in and there's a light in the center that you look at, and your job is, if a light appears on the left or the right side, you just follow it. This is very simple. This would be much the same as if this room was dark and my laser pointer was darting around here. All of you would be very good at following wherever that goes. As soon as all of you walk out on the street, you're very good at looking at cars, pedestrians, etc. So when you were doing this task of just looking at the light, your mind could quickly wander to, on the way home tonight, I have to go shopping, I have to stop at the milk store, whatever. And yet you would continue to make these kind of eye movements. So this is very simple, low-level behavior. Something sensory happens, a light appears, you do something motor, you look at it. And so I think my next slide might have my circuit again. Go backwards maybe. So to do that simple task, a light's going to appear on the screen. That's going to activate the green parts of this figure. And then you want to look at the light, you've got to activate the red parts, and away you go. And that's it. And all this other stuff in the brain just gets in the way, so we can ignore it. Okay, but that's when I gave you the really simple task of just follow the light. Now we're going to contrast that with another task that is going to be identical. There's going to be a center light that appears, and then a target's going to appear on the left or the right side, but your instruction now is very different. Now, when the light appears on the left side, you must look to the right, or if the light appears on the right side, you must look to the left. And this is not easy. This is very hard now, because you want to suppress and not look at the lights, which is very automatic. So there's a reflex trying to pull you over here. You want to not do that, and then do something more complex. Okay, so very similar what's happening in the visual world here, but the instruction here is very different. And now if you're thinking about going shopping this evening and getting milk, you're going to mess this up and you're going to make mistakes. And that's what I'm going to show you now. Okay, so if you were in the lab, or if you were over here with the eye tracker, we could measure what you did with your eye movements, and this is what you would do in this anti-saccade task. So time is going along, there's a red light in the center that says you're doing this very difficult task, then a target's going to appear, and you have to look the opposite way. And this is the position of the eyes of one of our subjects doing this task. So they're looking in the center, the target comes on, and they look away from the target in the opposite direction. That's the correct response. And sometimes you see this subject 3 times look the wrong way, they look at the light, and then immediately correct it and look the other way. So we see that even on these 3 trials where the subject made a mistake, they understood exactly what we were asking them to do, to look away, and instead they couldn't suppress this detour, this reflective urge to look at the light before they look away. So this subject, even on the mistake trials, they know exactly what we're asking them to do, they just can't get rid of this detour. And that's what we're going to measure and exploit. And the number of times that those mistakes come out are very indicative of problems in the brain. And I'm just going to speed through a final couple slides now showing you behavior of normal individuals across the lifespan from age 5 to, I think, 90 years. And then I'll just show you what a couple of neurological disease patients do. So all we're plotting here is this is time. This is when the light came on. And if you did it correctly, you would have some reaction time, and then your eye would go in the right direction, and this curve's going to jump up. And every time you make a mistake and you look at the light, we're going to measure the time when you look at the light, and it's going to push this curve down. And so these two curves are the distributions of reaction times of small children doing this task. They were between 5 and 8 years of age, okay? So they're horrible. You can see that they make almost as many mistakes as correct responses. But this isn't chance, because notice the mistakes happened earlier than the correct responses. So there's a race going on in their brain between, there's a reflex trying to pull the eye in one direction, and they are trying to suppress that and voluntarily do something in the other direction that's very hard. And these young children are not very good at that. Now, as we look at older children, so now these orange curves come from children that are 9 to 12 years of age, so less errors, more correct responses, and you see this part of the curve getting steeper, their brain's getting better at this reflex. But they're still making, early on, many mistakes. Now let's look at the teenagers. So these are children now between 13 to 16 years of age. Less mistakes, more correct responses. This part of the curve is getting steeper. So they're getting more voluntary control of their behavior, and they're able to suppress this reflex. And now we consider maybe giving them car keys. Okay, because now they're getting ready to make complex voluntary decisions. Okay, now we go a bit older. Oops, I went the wrong way. These are like late teens. Very few errors, lots of correct responses. The curve's getting even steeper. And now we let them vote, maybe give them access to alcohol, and we presume they'll never mix that with the car keys. Again, we go a little older, early 20-somethings, they're very good at this, very few errors, mostly correct responses. The brain is very machine-like now in producing these responses. And unfortunately, after about 25 years of age, it's downhill. And as we go all the way through the lifespan, out until the mid-80s, you can see this slowing down and the older people starting to struggle on this task. And so what I just showed you on this was just what normal behavior would look like. And I don't have time this evening to then show you what children who would have attention deficit hyperactivity disorder or fetal alcohol syndrome or autism, elderly patients maybe that have early Parkinson's, Alzheimer's, Huntington's disease, they start to deviate from this normal pattern. And that's what we really study in my lab. For the last few minutes now, I'm going to take a deviation from this. So I'm not going to show you any of what the different subjects, different patient groups do, but I can go into that in a lot more detail after informally if any of you are interested in. What I want to then now pose is the problem that this approach has, because that anti-saccade task is very complicated. So I'm giving you a complex instruction. Look straight ahead. When a light appears over here, you have to look over there. That's a lot of information to process. What we're really interested in is taking this task to very small children, maybe a year or two of age. They're not going to understand that instruction. They're not going to make anti-saccades. Likewise, we want to study the elderly, very advanced, and maybe some of them are heading towards amnesia, dementia. Again, they're going to struggle with the instruction of this complex task, and if they can't keep track of the instruction, they're going to have problems in this task. So we've gone to a new approach, and I'm just going to show you one example of this. We do a thing called dynamic viewing of video. So now we're going to hook the eye tracker up to you, and now all you have to do is watch TV. So I'm just going to show you what we would show to the subjects. Why is it this way? Just start it again. Okay, so imagine you're just viewing these clips. Ignore all the bouncing dots yet. So it's just a series of video clips glued together. There's no instruction. You can watch whatever you want. So we could put this in front of a little baby. They might watch some TV. We could put this in front of an old person. They like to watch TV. We can put this in front of anybody in between. They like to watch TV. They look around. Now, what you'll notice is these clips keep jumping from one thing to another. And we're looking in here for abnormalities that subjects are going to do with their eye movements. Let me just start this again. So now these dots are going to become important. This is actually where some human subjects are looking. And you'll see some of the subjects are coded in red, some of them are in blue. So there's a big difference between these groups. And you'll see that the blue and red dots deviate from each other every once in a while. Here's a good one maybe. You see the blues going one way, the reds going another. Then some other times they're all together and overlap. Here's a good one where the blues and reds start to separate. So now the jig here is, these blue and red dots are eye movements that were recorded from a group of undergraduate students at the University of Southern California. So these are normal young adults. And 6 of them were males, and 6 of them were females. And I hope you can appreciate just viewing those clips. Those subjects are doing some big differences at times. I won't tell you which one's which yet. I'll let you figure it out. I'll tell you at the end. In fact, maybe I'll just let it run again. But just when we look at gender, so these are all young 20-something subjects. And just by gender, they're doing very different things with their eyes. Had I shown you instead what old people would do compared to young people, there would be a big difference. If I showed you what Parkinson's patients do compared to normal old people, there would be a big difference. Anybody want to venture a guess here between the blue and red? Who were the males, who were the females? Yep. Red males, blue females. Who agrees with that? Some heads nodding. That's completely opposite. You'll notice what you were probably catching is the reds were following the female body figure more than the blues. Bit of a shock, eh? How ruthless females are when they're looking at other females. There's big differences, and you can see the males doing something different. So this is a ridiculously simple behavior. Probably all of you are going to go home tonight and watch TV, but we can carefully, by measuring your eye movements while you do that, decompose it and start to identify if the brain's functioning in a normal, healthy way, or whether there are abnormalities. And so I hope you can appreciate in these few minutes this little snapshot I gave you of how we can use this behavior and then study a whole variety of neurological, psychiatric